So Dr. Will tie this all together. Like you said, the question in everyone's mind is, are we there yet? So we'll hear some cool clinical applications.
Thank you for the introduction. Today I wanted to talk about the subcortical and cerebellum neural correlates of prodromal Alzheimer's disease with prolonged sleep latency. I am a psychiatrist, and Alzheimer's disease patients not only present with cognitive symptoms, but so many other symptoms, non-cognitive symptoms like motor, affective symptoms, anxiety, and everything. But I think subcortical regions and cerebellum are two most key regions that should not be under-recognized. And my presentation, through my presentation, I want to stress out the importance, clinical importance. I have no conflict of interest to declare. This is the background of our study. There have been reports of detrimental effects of prolonged sleep latency and dementia, and this was the Korean group reported in 2018. When compared to short sleep latency at baseline and follow-up in cognitively normal participants, when compared to them who changed from short to long, and long at both baseline follow-up, they were both at high odds of cognitive risk decline, cognitive decline risk. And there was a positive correlation between mean sleep latency and amyloid beta deposition, especially significant in frontal regions. Actually, frontal regions are, along with the precuneus and the posterior cingulate cortex, they are early regions of amyloid beta deposition. So sleep disturbance and amyloid deposition in early stages of AD, and we should focus on that. And as for the why we are focusing on subcortical regions and why I picked locus coeruleus in particular was that in 2021, Jacob's group reported that locus coeruleus may be an incipient Alzheimer's disease marker because it's a site of early locus coeruleus tau deposition. And it may start early as age as 30 years of age, and its importance as an incipient biomarker was stressed. As you all know, locus coeruleus is the blue spot is very important for regulating sleep-wake homeostasis. And it's norepinephrine noradrenergic firing. It's usually silenced in non-REM sleep and REM sleep, and locus coeruleus is important for maintaining sleep-wake homeostasis. And we thought that there may be a link between locus coeruleus, early Alzheimer's disease pathology, and sleep. As for the cerebellum, previous studies reported that like a lower cerebellar anterior lobe volume in early onset AD and early stages of AD patients presented with posterior lobe changes in posterior lobe of the cerebellum. And as the disease progressed, the anterior lobe was involved, and reduced white matter interiority in the posterior lobe in MCI was also reported. And as for functional correlates in MCI patients and also in Alzheimer's disease patients, like functional connectivity changes were correlated with clinical measures like semantic fluency and MMSE scores. So our hypothesis was that we aim to investigate how amyloid deposition and prolonged sleep latency interact to generate structural and functional alterations of the subcortical structures and cerebellum in MCI patients. And we hypothesized that amyloid-positive amnestic mild cognitive impairment patients, who we think is ultra high risk of conversion to Alzheimer's disease dementia, and who present with prolonged sleep latency will display distinct alterations in subcortical structures, especially in the brainstem, thalamus, and the hippocampus. And we postulate that the locus coeruleus, hippocampal, thalamus, cerebellar functional connectivity will be disrupted in patients with prolonged sleep latency, which will be caused by interaction between amyloid deposition and prolonged sleep latency. 60 amnestic MCI patients were recruited, and all amnestic MCI patients met the Peterson's criteria of MCI and displayed objective memory impairment scores of 1.5 standard deviations below the sex education and age-specific normative means in at least one cognitive domain, testing for episodic memory. And all the participants were identified as amyloid beta positive and filter metamol positive emission tomography acquisition section. And as for the assessment sleep latency, we adopted the Korean version of Pittsburgh Sleep Quality Index. And the second item of PSQI asks the patients, how long in minutes has it taken you to fall asleep each night? And prolonged sleep latency was defined as those with sleep latency of 30 minutes or more. And this is the summary of data acquisition and data analysis we've performed. Voxel-based morphometry and surface based analysis of cortical thickness and brainstem thalamus hippocampal volume analysis and resting and functional connectivity analysis. And this is the result. So amnestic MCI patients, there were no demographic characteristic differences between the two groups except for sleep latency. And as for the volumetric analysis, brainstem volume was significantly reduced in prolonged sleep latency group, and there was a trend for reduced left thalamic volume in prolonged sleep latency group. And as for the resting state functional connectivity analysis, there were seed regions we chose, like anterior cerebellum network, posterior cerebellum network, locus coeruleus, and left hippocampus, right hippocampus, and bilateral thalamus. And there were group difference of locus coeruleus functional connectivity with right frontal cortex and left thalamic functional connectivity differences with left occipital cortex. And there were increased locus coeruleus functional connectivity with occipital cortex and left hippocampal connectivity with frontal cortex in prolonged sleep latency group. And there was a sleep latency group by posterior singular cortex standardized update value ratio interactions in anterior cerebellum network functional connectivity with right frontal cortex and parietal SUV interactions in posterior cerebellum network with right temporal cortex. These are the discussion points. As for there was a significant reduction of the brainstem volume and a trend for the reduction of left thalamic volume in prolonged sleep latency group. There were previous results where the brainstem volume and locus coeruleus volumes were compared between cognitive normal patients, MCI, and Alzheimer's disease patients, and there were reduced brainstem volume and locus coeruleus volume as the disease progressed. And when non-converters and converters were compared, there were reduced brainstem volumes and locus coeruleus volumes in converter group. So there are changes in subcortical and subcortical changes in early stages of Alzheimer's disease. And in a recent study in Alzheimer's Research and Therapy, Grover-Rivera's group reported that the insomnia patients who are cognitively unimpaired had reduced bilateral posterior single cortex precuneus and right thalamus and left thalamus volume. So there may be changes early as like in cognitively unimpaired groups. And in our results, the locus coeruleus functional connectivity with the insular cortex was reduced in prolonged sleep latency group. And in a recent study reported that the LC and noradrenergic system and salience network were closely connected with a reduced functional connectivity between the locus coeruleus and salience network, resulting in impaired attentional and executive function in older adults. So the prolonged sleep latency group in our subjects may have this impaired network, a crosstalk between locus coeruleus functional connectivity and insular cortex. And in our result, there was a reduction of the locus coeruleus functional connectivity with frontal and temporal cortices. Locus coeruleus and frontal cortex are suggested to play an integral role in synaptic plasticity And control of attention and response inhibition was mediated by the local serous projections to the dorsomedial prefrontal cortex and the ventrolateral orbital frontal cortex. The locus serous functional connectivity with right hippocampus was reduced in prolonged sleep latency subjects. And a recent study reported that the norepinephrine antagonism prevented dopamine-mediated long-term potentiation in cornus amnus, one region of hippocampus. The LCNE system is connection to hippocampus, and noradrenergic signaling may be disrupted in prolonged sleep latency subjects, resulting in reduced locus serous functional connectivity with the hippocampus. Increased locus serous functional connectivity with the occipital cortex was observed in a prolonged sleep latency group. In a previous study on patients with chronic insomnia disorder, insomnia duration was closely associated with the locus serous occipital cortex functional connectivity. And this was supported to be a representation of a hyperaroused state of the visual cortex. And in a functional connectivity study adopting a machine learning algorithm in classification of subjects at early stages of AD, occipital cortex was identified as a resting state functional MRI hub node in MCI converters. So we thought disruption of visual processing and occipital functional connectivity may start from an early stage of Alzheimer's disease, and the brain may attempt to compensate for the failing function by increasing the locus serous functional connectivity to the occipital cortex. So hyperarousal and result of prolonged sleep latency may be a phenotype of this underlying compensatory process. And left thalamic functional connectivity with both occipital cortices were decreased in prolonged sleep latency group. And previous studies reported that alterations of the alpha rhythm originating from the thalamocortico-occipital circuit in the early stages of AD. And a crosstalk between thalamus and cerebral cortex is integral to the generation of slow wave activity during non-REM sleep. So disrupted sleep in prolonged sleep latency subjects may have disrupted thalamo-occipital circuit, and whether this was a synergistic effect with AD pathology or independent effect remains to be elucidated. Yes. Left hippocampal functional connectivity with both frontal cortices were increased in our prolonged sleep latency subjects. This is concordant with a previous study where insomnia subjects presented with increased hippocampal functional connectivity to frontal cortex. And this was correlated with insomnia severity scores and sleep efficiency. As for the cerebellar network, there was a sleep latency group by posterior single cortex SUVR interactions in the anterior cerebellum network, showing decreased anterior cerebellum functional connectivity with frontal cortex. And there was a sleep latency group by parietal amyloid beta SUVR interaction in the posterior cerebellum network. And there was a previous study where the cerebellar functional connectivity to the cortex was compared between healthy control group, mild cognitive impairment, and Alzheimer's disease dementia. And there were differences between the two groups. And I think there may be changes in the cerebellar network, even in early stages of disease, and those with sleep disruptions. The role of cerebellum in sleep is largely under-recognized, and a recent study reported an important role of the cerebellum in the generation of neocortical sleep spindles. So subjects with sleep disorders displayed decreased cerebellar volume, and increased water diffusivity during sleep was noted predominantly in the cerebellum in humans, suggesting the indispensable role of the cerebellum in the glymphatic system during sleep. These are some of the limitations of our study. The sample size was small, and the study was cross-sectional, and the assessment of sleep latency was self-reported, and comparisons between amnestic MCI subjects and healthy controls were not made. And because since the research on the LC and cerebellar functional connectivity in aging population is still in infancy, so a consideration for physiological aging effects of the LC and cerebellar functional connectivity was absent, and the inherent limitations of functional connectivity studies and maybe our limitations to consider. And I recently expanded my research to the impact of apolipoprotein E4 genotype on locus functional connectivity in preclinical Alzheimer's disease and also the sex-related disparities. I think the importance of cerebellum in subcortical regions and should not be under-recognized and we should expand the research in neuroimaging area. Okay, this is my team. I'm under Professor Lim's lab, and it's an honor to speak to you. And then thank you for your attention. Thank you.